Yes, level biology 9700, topic 1, cell structure. This is the second video on this topic, cell structure. In the video 1, we had discussed about the syllabus. And secondly, we had discussed about light microscope and the magnification. Now, in this video, we are going to discuss about the electron microscope, which is the other type of microscope in which instead of light, the electron beam is there to produce the image of any specimen. So the different parts of electron microscopes are there, that is the electron gun and electron beam. Electron gun will produce this electron beam and magnetic lens. Different magnetic lenses are there, which will help in the production and magnification of the images on the screen or in the photographic plate. So these are the different parts which has been shown over there. Now I would like to move towards the different parts, the functions of different parts of this electron microscope. So first of all, you can see over here on screen, that is the electron gun and anode, which will produce a beam of electrons. Actually, this electron gun has a tungsten filament which will produce this beam of electron. So this beam of electron will pass through a vacuum, and then there is condenser electromagnetic lens, which will direct this electron beams onto the specimen. So it will move in the downward direction towards the specimen, and this electron beam will pass through this specimen, and then there is an objective electromagnetic lens. So the objective electromagnetic lens will produce an image which will be projected by the projector electromagnetic lens. So in this way, it will focus the magnified image onto the screen. So there is screen or micro photographic plate over there which will show the image of this specimen. So it means there are different types of electromagnetic lenses which are used to produce this image of the specimen. So in this way, in the coordination of all these lenses, the electron beam which is produced from the electron gun, all they will collectively produce an image of the specimen on the photographic plate or on any screen or computer screen as well. So it can be produced over here. So it depends on the type of the electron microscope. So in this way, image is produced. So there are some limitations in the production of the image by this electron micrograph. That is, first of all, which I had shown you in the previous slide, that is the vacuum. This electron microscope only works in the vacuum. If there is humidity, it will not properly work. It cannot be operated. It cannot be, you can say, the working condition. So vacuum should be there when we are going to produce an image by the electron micrograph. Another thing, the live specimen cannot be observed. Live specimen cannot be observed. Only the dead specimens are available to observe under that microscope. So as the presentation power of the electron beam is very low, the object should be ultra thin. So ultra thin sections of the specimens are required to observe them under the microscope. So for this purpose, the specimen is dried and cut into ultra thin sections before observation. If they are not ultra thin, if they are not dried, they are humid, or they have some water inside or moisture inside. So what will happen? The proper production of the image will not take place. So the specimen should be completely dry, expensive to build and maintain. So, you know, this microscope is large in size. It needs a large room to work with, and its maintenance, and the persons are needed for its maintenance and research, requiring researcher training. So the trained persons can only operate this type of microscope. So this type of microscope is a large, cumbersome, extremely sensitive to vibration and external magnetic field. So it means any external electronic magnetic field can disturb the formation of the image. So in this case, these are the different limitations which should be considered before producing an image. Beside this, there are, you can say, many benefit of this use of this electron microscope, as for example, magnification and higher resolution. This they have. I would like to write over here magnification. They have high magnification and high resolution. So it means electron rather than light waves are used. It can be used to analyze structures which cannot otherwise be seen. So more precise or more detailed structure of these specimens can be observed under this microscope. So there are, you can say, it can be applied in various applications. So electron microscopy has a diverse range of applications in many different fields of research, including technology, industry, biomedical science, chemistry. Examples, so you can say other examples can be included semiconductor inspection, computer chip manufacture, quality control and assurance. So diverse application. It has diverse applications. So it can be applied in various fields of life. High quality image, the images which are number 3, high quality image. So high quality images can be produced, but you know, the persons who are working with this electron microscope should be trained, should be highly, you can say, qualified for this purpose. Then this microscope is very beneficial for the use in the laboratories or in different fields of life. Now next, I had discussed that it has high resolution power. The electron microscope has high resolution power. What does it mean? What is resolution? So the resolution of an optical microscope is defined as the shortest distance between 2 points on a specimen that can still be distinguished by the observer or camera system as separate entities. So resolution is the, you can say, a shortest distance between 2 points on a specimen. As for example, we have 2 dots or 2 cells. As for example, they have very shortest distance. We cannot observe the shortest distance by our eye or by watching it under the microscope. But the resolution, if the microscope has higher resolution, then rather they are very close to each other. They will be shown as different dots or different cells. They are very close, they are joined together, but still there is a distance between them, a shortest distance between them, due to which we can observe them at 2 different cells. So in this way, the resolution of electron microscope is always higher than the light microscope. But you can see light microscopes are very easy to use in the laboratories, and especially for the students, it is very because there is no technique required to use them. Only if you know the parts of, and once if your teacher has taught you how to use the microscope, it is very easy for you to use that microscope, because you have paper 3 in which you have to observe the some specimens under the microscope to for your educational point of view. So resolution is that shortest distance, or resolution is that you can say which allows us to see objects as separate from one another. They are, if they are closely closed together, but they will look like, as they are not closed, they will be different. So it means resolution will help us to observe the any specimen very clearly. So electron microscopes are of two different types, which we have to discuss in our syllabus. So there are different types of electron microscopes, but we have to discuss about only two. That is the transmission electron microscope and scanning electron microscope. So this transmission electron microscope passes through a very thin section of the material. So very thin section is required for it, and then it will pass through, but it will show some parts of that image. It will show the internal structure. It will show the ultrastructure details about some parts through which the electrons beam will pass. The electrons will pass. So only those parts will be shown through which the electron beam will pass. So they will be visible. So it show the internal structure. So on the other hand, this scanning electron microscope, or SEM, a narrow beam of electrons is scanned in a series of lines across the surface of the specimen. So mostly it will show the surface area, surface region of the specimen. So the electrons that are reflected or emitted from the surface are collected by a detector and converted into an electrical signal, so which is used to build a 3D image. So it will show the outer 3D image, or three-dimensional image, line by line on a TV screen. So it can only be observed on TV screen. It will pass through it. It will produce the internal structure, but it will externally, it will show the 3D structure because it will show the surface parts. So so in this way, it will show only the external area or external part of the any specimen. So this is the difference between these two different types of electron microscopes. With the help of electron microscope, we can have the detailed structures of different cells. So here you can see on screen the ultrastructure of an animal cell. You can say the detailed structure has been shown. Otherwise, if we observe any cell under the microscope, then we can only see the cell wall, the nucleus, and the cytoplasm over there. But if we observe any specimen in the under the electron microscope, then we can observe the ultrastructure of that specimen. So ultrastructure of a cell is a detailed microscopic structure of a cell. Its components, particularly those which are visible under the electron microscope. So it can it helps us to arrange, it helps us to show the arrangement of the different organelles, membranes, and molecular complexes within the cell. So it is a deeper look into the cell's architecture beyond what is visible with a light microscope. So it means it can only, the detailed structure can, or the ultrastructure can only be seen with the help of electron microscope. So here you can see the whole cell of an animal. So different parts, you can see the vacuole vesicles, which are very close to the cell surface membrane. And plasma membrane or cell surface membrane is the outer membrane of an animal cell. And in the center, you can see a big nucleus. Inside the the nucleolus is also visible. So from the nuclear envelope, you can see originating the endoplasmic reticulums, and you can also see the rough endoplasmic reticulum as well the uh, smooth endoplasmic reticulums. On rough endoplasmic reticulums, you can see the ribosomes are attached. So these ribosomes are attached on the endoplasmic reticulum. So due to which the uh, endoplasmic reticulums are called as the rough endoplasmic reticulums. The ribosomes are also available in the cytoplasm. They are freely available in the cytoplasm. So lysosomes are there. The lysosomes are also part of the cell, which we will discuss in detail. And you can see the vesicles, and you can see the mitochondria, which is you can also observe the membrane of this mitochondria. You can observe the Golgi apparatus, which is formed from the endoplasmic reticulums, and you can see the centrioles, which are only present, a pair of centrioles, which is only present in the animal cells, and microtubules are also there, which help to maintain the structure of the animal. And then you can see also the free ribosomes are there. So in this way, the detailed structure of the animal cell. Now we will move towards the ultrastructure of a plant cell. In the next video, in the next slide, you can see the ultrastructure of the plant, and outermost layer you can see the cell wall, which is absent in animal cell. Plasma membrane is there. So central single large vacuole is there. Again, mitochondrions are there. Mitochondria are there. The chloroplast are there. Plasmodesmata, which are the pores into the uh, between two cells into the cell wall, and lysosomes are also there. Ribosomes are there, and you can see nucleus has nucleolus, and you can see the endoplasmic reticulum is also there. Smooth endoplasmic reticulum, rough endoplasmic reticulum, both are visible over there. So Golgi apparatus, mitochondria, central vacuole. We will discuss all these parts one by one in the next videos, inshallah. So here you can see the different parts which has been shown. All they are visible under the microscope. Electron microscope. The detailed vision is only under the electron microscope. Now we will move towards the next video of this slide. Nucleus has been shown over here. The nucleus is the major part of the cell which control all the activities. You know very well about the nucleus. Now you can see its detailed structure. You can see the nuclear membrane is there, which is porous in nature. It is double membrane structure. A double membrane structure, nuclear pore, nuclear which are present in which controls the. So nuclear membrane also act as partially permeable, which allow only some molecules to pass through it. Now you can see nucleolus is there, and then you can see nuclear envelope, nucleolus, and chromatin material is inside. Chromatin material is, you can say, this is DNA and chromosomes. So it is not visible till there is cell division. So at the time of cell division, it is condensed, and the chromatin material take the shape of the chromosomes, and they take the prominent shape, and you can also see the DNA present inside the chromosomes. So in this way, you can see that the nucleus is a very organized part of the cell, and it is very important part which control the activities. So nucleoplasm is also there, a jelly-like substance which is present inside. So you can see from the nuclear pore or the nuclear membrane, the endoplasmic reticulum are originated. On them, the ribosomes are also attached them, due to which we say they are the rough endoplasmic reticulum. If the ribosomes are attached on them, then they are called as the rough endoplasmic reticulums. Otherwise, they are smooth endoplasmic reticulum. So I have told you about nucleus. Now here I'm going to read about this. Nucleus is the largest organelle. It is surrounded by two membranes known as the nuclear envelope. Outer membrane is continuous with the endoplasmic reticulum. So the nuclear envelope has many small pores called nuclear pores. So these allow and control exchange between the nucleus and the cytoplasm. Examples of substances leaving the nucleus through the pores are messenger RNA and ribosomes for protein synthesis. Examples of the substance entering through the nuclear pores are proteins to help make ribosomes, nucleotides, ATP, and some hormones such as thyroid hormone T3. So these are the, I already told you that it will allow some molecules to pass through it. So the entering substance entering through the nuclear pores, which will be allowed to pass through, they are the ribosomes because they have to do the protein synthesis. Nucleotides for protein, ATP, the energy, adenosine triphosphate, energy is necessary. So some hormones, thyroid hormones, which are called as T3 hormones, so they can also pass through this nuclear membrane. So it was all about the second video on cell structure. Please like, share, and subscribe my channel to watch more videos. Thanks for watching.